new news slide set on plant growth after that. So the one thing that you probably didn't really notice per se, if you were looking at cells under the microscope yesterday, was the vacuole, because you have to use a really special staining technique to get it to pop into view really nicely. However, if you did see under the microscope that, gee, it seems like the middle part of the cell, there's nothing there and everything's squished around to the edges, that's because the vacuole was in the way and you just can't see it without a special stain. And so very often, if you have a really well-watered plant tissue that you're looking under the microscope, that's what it looks like. It's like the middle looks devoid, like there's nothing there, but all the organelles are like right around the perimeter of the cell. So the tonoplast is the name of the membrane that surrounds the vacuole. They tend to call it the central vacuole because it does tend to be more in the middle of the cell. It's very big. It's bigger than the nucleus, which the artist has drawn correctly here. And actually, when you think about the vacuole, the number one component of the vacuole is water, hopefully. In a well-watered plant, you want water in there. If it's a wilted plant, not so much, but you want the vacuole to be well watered. And so here, again, you can barely see any outline to the vacuole. It just looks like a devoid space. And again, you can see the nucleus is much smaller. And then here, this picture on the bottom right, that's the electron microscope picture where you can see, again, the vacuole just takes up a big part of the cell and everything else seems squished off to the side. So that's important because if that vacuole is filled with water, like especially in that lower right image, then what's happening is the water pressure inside the vacuole is great enough that it's pressing against all the organelles, which presses against the cell membrane and the cell wall. So you get a structurally strong area in that plant tissue. And so if you have a well-watered plant, I'm sure you've noticed this if you have plants at all, then they leaves stay nice and fully expanded. You have the stem upright. Everything looks lovely. Wilted, of course, everything starts to droop. And so in large part, it's because if you have a wilty plant, the vacuole is devoid of water and everything just kind of relaxes and wilts. Okay, so that's a big difference compared to animal cells is that we don't have that big central vacuole like that. We have bones to help us fight gravity and have some structural integrity. Okay, let's zoom out from cells and get a little bit more macroscopic. Let's get into kind of the overall body of the plant, and then we'll look at some key parts. So here, I mean, it's really quite remarkable. Plants live a really interesting lifestyle in the sense that more than half of them is buried underground. And so most plants that you look at when you're out walking around or hiking in the woods, what you see above ground, there's actually more below ground. Usually the root biomass is the larger portion of the plant. We just don't see that with our eyes because we don't look into the soil on a daily basis. But the root system can be quite extensive. And so more than part of their body is underground in the dark, surrounded by soil and all kinds of interesting organisms. And then above ground, they have to fight gravity. You have all kinds of other issues in play that they have to experience and figure out what to do. So generally, when you look at the plant body, you can see a couple of things going on here. So when you got your seeds going yesterday too, right? They're germinating. So the first thing that will come out of those seeds is actually the radicle, which is the embryonic root. And you're going to see that you have a root apex or a root tip. And the root apex, they call it the apex or the apical. Well, they call it the apex, which means it's the tip. And then you see there's a shoot apex as well, because there's a tip to the shoot. And then in that region, it's what we call the apical meristem. I'll just go forward for a second into this slide. So the apical meristem, it's the shoot apical meristem and the root apical meristem. So there's two. And that helps the plant grow longer. Roots grow longer, shoot grows longer. All right. It's a very precious area of the tissue of the plant because that's where you have really active cell division going on a pretty regular basis. You want those cells to be very healthy and active. All right, so you have that going on. You also see a root cap that covers the very tip of the root. And that's important because the soil is very abrasive. And having that root cap is protective. That root cap is composed of a bunch of cells that just kind of surround the root apex. They slough off over time, but new ones are produced. So it's a very dynamic area. And it's helping to protect the precious root apex with all those active cells going through cell division. You see there's root hairs as well. Hairs are very important for water absorption. So they're very fine and thin. We'll look at that a little bit more. Depending on the plant species, you can see that there might be a central taproot where what that means is there's usually a really obvious central root that's very big. And then you might see some lateral roots that are smaller. So you might see that kind of root system. But we'll look at roots a little bit more detail in a little bit. Okay, because there's different morphologies of root system. They're just drawing one example here. All right, what else do we have going on? We have where you see the leaves attaching to the main stem here. If you look in the little crook or crevice there, you often see an axillary bud. You do need to know that term, axillary bud. You can see there's a little bud of tissue there. So the axillary bud is another area of really active cell division sometimes. All right, sometimes, not always, not nearly to the extent that you see with the shoot apex. So the axillary bud, if that decides to grow, you can get new leaves, new branches, depending on the species of plant that you're looking at. So we call that the node region of the stem. Anywhere where you can see on the stem, oh, there's an axillary bud, we call that the node region of the stem. And then here they've also labeled the internode. That's just the space in between two nodes. Inter means between. So internode just means it's the space between two nodes. So I'm not too worried that you know that term. But knowing what the node means, that's where you would find the axillary bud and usually leaves coming off of that or a branch perhaps. Okay. And then the artist has peeled away part of the outer portion of the stem, and we're getting a peek inside at the vascular tissue. So we'll look at that as well. So the apical meristems, the shoot apex, root apex, and you can see that the root system has lots of apical meristem regions at the tip of each root that you see there. So lots of that going on. And then they're also trying to show that you have some lateral meristems that they put in red here. So the lateral meristems, I like to look at it in cross section, like over here while they took the section out. We're going to look at lateral meristems in more detail, but the lateral meristem, when you take a cross section like that, it does form a ring of tissue like that. The lateral meristem, again, active area, anytime you see the word meristem, that's active areas of cell growth. Lateral just means that when that meristem tissue is very active, it helps increase the girth or the diameter. So if you think about giant redwood trees or the sequoias, giant sequoias, you definitely have very active lateral meristems over the lifespan of those types of trees to create such huge tree trunks. So lateral meristems. So you're starting to see that there's discrete regions in the plant body where there's really active growth, which is necessary. Plants can keep growing and growing and growing. It's really dependent on the environment they're exposed to, how much they can do over their lifespan. Okay. So apex, another word for that is apical. That's why they call it the apical meristems. We tend to say apical meristems rather than apex meristems. So. All right, and then lateral meristems are being shown here a little bit more detail. So the lateral meristems, one example is the vascular cambium, which we'll get into in more detail when we get into roots in more detail. But you can see it here. The vascular cambium, if you look at this bigger picture here, and you see that obvious break here where you have this deeper pink swath of tissue, and then there's this darker band, and then the lighter pink. That darker band that separates those two parts, that's your vascular cambium. And we'll get into this in more detail as we get into roots in more detail. So that is an area of really active cell division. And again, it's making the tree trunk bigger in diameter. And then outside of that, you have what's called a cork cambium, which we'll get into in more detail. But the cork cambium is further out. It's this line that maybe you can see. It's hard for me to draw when everything's so red. Line here. And that's your cork cambium. So this cork cambium is actually making more bark tissue. So cork cambium is for bark development, okay, to get your bark growing on the tree trunk. So we say those produce secondary tissues because, mainly because we call primary tissues things that are produced by the shoot apical meristem, the root apical meristem. We say that's all primary growth because it leads to the length of the plant. Whereas the secondary growth leads to the width of the plant, okay? So that's a little bit of jargon, unfortunately, you have to think about. This image, by the way, is showing the progression of growth. So when you have a younger tree, and then the arrow is pointing to a little bit older, and then finally you see here it's looks like a couple-year-old tree. You can see the tree rings. So it looks like we don't count the middle, but that's a tree ring there, and another, so it's 1 year, 2 years old. So you'll look at some of these in lab later.
You said the vascular cambium was that lighter area.
So it's the area, the vascular cambium is just a line, it's a row of cells, a band of cells, I guess, that goes all the way around. It's this area that. Yeah, so, and we'll see some better pictures of this, and you'll get to see this in lab too. And we, we're going to be covering root stems and leaves in more detail. Right now, I'm just kind of introducing some ideas here to get comfortable with. Grasses are kind of fun because they have what we call an intercalary meristem, and that's why you always have to mow your lawn. It's due to the intercalary meristems. You mow your lawn, and the intercalary meristem is at the base of the leaf blade. You missed it when you mowed, and those cells just go, yeah, fine, I'll just produce a new leaf. And so the intercalary meristems are very common in grass species, and that's why you got to mow. By the way, the lovely smell of fresh cut grass is actually the plant yelling out, help, I'm being damaged. So it's kind of a bummer, but yeah, it's under the impression that it's getting eaten. And so that lovely smell is actually a secondary metabolite, and it's trying to warn others, I'm getting eaten, you're probably next. So, but to us, to our nose, it usually, most people find that very pleasing, even though the poor plant is thinking it's under attack. Okay, tissues. We need to think about tissues. There's different tissues that make up the plant body. Of course, they had to make them some weird spelling and hard to pronounce. Parenchyma is one type of tissue. In fact, if you were to say, what's the most abundant tissue type in the plant body? Parenchyma wins. There's more parenchyma tissue in the body of a plant than any other. They're living cells. They don't look like a whole lot. That's a section of parenchyma tissue, and each of those cells is parenchyma cells. They're living. They're pretty thin. So they just have a primary cell wall. They're pretty thin. There's so you're going to see a lot of parenchyma cells in lab when you're looking at different parts of the plant body. So they kind of have these many sides to them. If you look around the perimeter, they fit together pretty nicely with each other. CW is cell wall. Their cell wall is pretty thin. Usually, if you say thin, that means it's just primary cell wall, not a secondary cell wall. By the way, if you are using my PowerPoint, you'll see that I have extra words in the notes section. Okay, so those are notes that I used to put up in the PowerPoint, but I didn't want students writing that all down. Which reminds me, it's good to take notes, and I'm fine with that. That's a good strategy. However, I do caution you that if you write down everything that's on my PowerPoint, then you can't really hear what I'm saying. And I mean, it's a known fact, you can't do both. And so I would prefer that you find a way to not write all this down. It's all provided to you. Just take extra notes of the stuff I'm saying when I explain it, because you really, I mean, the neuroscience shows you can't write and listen at the same time. So just be careful about that. If you're typing, you can put stuff in the notes section, but I don't know if you've seen the new neuroscience, but they're saying now handwriting is better than typing. Have you heard about that? They're saying you learn better if you handwrite. So give that some thought too. But anyways, okay. Uh, Here's some more tissues. So next week, I believe you'll be looking at parenchyma tissue, another hard word. Parenchyma tissue is full of parenchyma cells. And so they you can see that they fit together kind of oddly compared to parenchyma cells, like there's some spaces in between the cells. They're living cells, but what we find is their primary cell wall is a little bit beefed up, and that creates a little bit of space. When you look at it under the microscope, it looks like they're kind of spaced out a little bit amongst each other. This particular image is very dramatic, where you can see that there's lots of space, what looks like space, but that's you get cell walls that are thickened up and they just kind of bump against each other like that. And so I think they also will say that it can look uneven, the cell walls look uneven. So the cells next to each other don't perfectly like hug each other. There's like little gaps and unevenness. And so that's something you would learn to train your eye to. You look for that kind of pattern. So in lab next week, probably the best example of this is that we'll have available is celery stalks. You know how it's ribbed on the edge of the stalk. And if you've ever bitten through celery, although celery is not my favorite thing to munch on, but sometimes when you bite through it, you get those strings that you don't completely bite through it. Those strings of tissue, that's parenchyma. It's tough for your teeth to bite through. You have to be pretty aggressive when you bite celery stalks. And so that ribbing pattern that you see on the stalk of celery, that's clusters of parenchyma cells. And so if you can see here in this partial image, I believe this is a celery stalk, and you can see how it kind of bumps out like this on the perimeter. And all that bumpy area is full of those parenchyma cells. Okay. So they are strong, they're alive, but because their primary wall is thickened up, it's like the celery stalk, they can be difficult to chomp through that cleanly. Okay, so parenchyma, parenchyma, they're both alive. And then you have sclerenchyma tissue, which is composed of sclerides and fibers. So you have sclerenchyma cells. They are thick. Scleroderma cells are thick, which means their secondary cell wall is really thick, I should say. So if a cell has a secondary cell wall, is it alive or dead? So this is from Monday. Remember, cells can have primary cells and secondary cells. Every cell is born with a primary cell wall. What about a secondary cell wall? When that cell forms a secondary cell wall, is it alive or dead? Dead. It's dead. Which may sound like, well, why would you do that? But there's a reason. Plants are doing that for a reason. And so in this case, sclerides are being shown here. There's no easy way to define them visually because they can look so different from each other. So like these star-looking cells, it's kind of a weird star shape. Those are a bunch of sclerides. But these are also sclerides too. There's several of these funky looking, sometimes they're referred to as stone cells, right? So sclerenchyma cells are dead. And they typically, because they have that thicker secondary cell wall, they usually help with structural support in that, wherever they're found in the plant body. So what you're looking at are what are called sclerides, a certain type of sclerenchyma cell that we call sclerides. And again, they can have different morphology. The best example I can give you, you've experienced sclerides when you bite into pears and eat a pear and you feel that grittiness on your teeth as you're chewing the flesh. That's a bunch of sclerides that you're eating, okay? So that's one reason why pears are so high in fiber and they're good for you in that regard for fiber. Okay. Sclerides tend to be scattered. So when you eat that pear, the sclerides are throughout the whole fleshy part of the pear. Okay, so they tend to be scattered around in that section of tissue in the plant body. And the only way that they can define them visually is they say, well, the cells are as long as they are wide. That's pretty much how they define it as what it looks like visually. So they're about as long as they are wide. Okay, so that's one kind of sclerenchyma cell. There's another kind called fibers. So sclerenchyma cells are sclerides and fibers. Fibers, you can define them. They are pretty much the same looking. They're always longer than they are wide. I like the picture on the left because it shows how the clusters of fibers. So you see all these pink cells. That's a cluster of fibers. Okay, whoops. And you can see all that pink. That's the cell wall. And then the little white dot in the middle, that's the dead cell. So when a cell is growing and let's say it's destined to become a fiber, what happens is the plant says, okay, you got to become a fiber. So then it starts laying down that secondary cell wall. And then by the time it's mature, it gets rid of all of its inner contents, the cytoplasm, all that gone. All right. And it's just a hollow cell that's beefed up with this really strong secondary cell wall. And so if you have a cluster of those together, that's a very strong area of the tissue and can be very helpful for the plant to stay upright. This is in longitudinal section. You can't really see a whole lot that's going on there in that view really, but you'll see some of that in lab as well. Okay, so what tends to happen is a plant will decide to produce fibers right around vascular tissue. We're going to see that a lot. That's a very common strategy. So the vascular tissue in a plant is kind of like our blood vessel system. All right, very precious part of the plant body because it's allowing flow of material through the whole plant body, just like our blood vessel system. And you want to protect that. You don't want a bunny rabbit coming along and chomping away all the vascular tissues. So they tend to put fibers, cluster these fibers all around the vascular tissue to protect that precious vascular tissue. We'll see some pictures of that. Okay, so here, now we're going to get into what they call complex tissue, which just means that it's composed of more than one cell type. So far, we've only been talking about single things, but now we're getting into more complex thing, which mainly we're going to be thinking about the vascular tissue as an example of this. All right, so it's kind of interesting. The artist drew the vascular tissue kind of like how artists do for the animal body with the veins and blood vessels and the red and blue is very typical in a human drawing or animal drawing. But anyways, okay, so we have vascular tissue. Vascular tissue goes throughout the whole plant body, all the way from the root apical meristem region area all the way up into the leaf and the leaf veins. All right, so we have that vascular tissue throughout the whole plant body, and it's composed of a couple of things here. We see xylem is one major type, and then phloem is the second major type. Okay. So what are xylem? Xylem tissue, depends on the plant, but some plants, when you look at their xylem tissue, they have what are called vessels, okay? And vessels are longer than they are wide. They're dead at maturity, which means they have a secondary cell wall that's pretty beefed up. So that picture is showing some examples of vessels. All right, it depends on the plant species, how wide it is. Some are a little bit more wide than others. But yes, definitely a thick secondary cell wall. If you look at the the vessel, not xylem, in general, in longitudinal, you'll often see this interesting pattern, kind of the spiral thickening. But anyways, you see that they're open at the ends. And now you have to imagine those vessel elements, they just stack on top of each other, one on top of the next, on top of the next, on top of the next. And then that forms this big, long, open conduit of, in this case, vessels that go through the whole plant body. All right. And then you have to imagine these are empty, no cytoplasm in there. It's just empty. But that can allow, the job of xylem is to allow for water movement through the whole plant body. And so if you think about it, it kind of makes sense that xylem doesn't have any cytoplasm because that allows for water to move through the plant body really efficiently. If you just have a hollow tube, it's like sucking pop up through a straw. It's very easy. Okay. And so it's actually a good strategy to have a bunch of dead cells because it allows for very efficient flow of water. Okay, so vessels are one type of xylem. Then there's another type called tracheids. Tracheids are more primitive or more ancient. Okay. So some plants that first developed a vascular system, they started out with tracheids. That was their first go at it in the evolutionary process. We see they have tracheids. So these are more primitive. Vessels are more advanced. So the flowering plants that are the most advanced species, they have a bunch of vessels. They also have tracheids. They have a mix, and we'll talk about that later. Okay, they tend to be tapered at the ends. They're skinnier than vessels for sure. But they're the same as the vessels in the sense that they're dead at maturity. So they have a nice thick secondary cell wall. They're empty, no cytoplasm or anything else. So again, very nice for water transport through the whole plant body. So you have to imagine these tracheids, again, they're stacked one on top of each other and they form those long conduits all the way from the root all the way to the leaf veins. You're looking at some of the veins is composed of the xylem. Then if you take like a tree trunk and you cut it in cross section, that's what they're showing down here. You'll see that some of the vascular tissue has this, I don't know if you can see that, those lighter like bands or rays they call them. That's actually going through a bunch of the xylem in a tree trunk. Most of what you're looking at there is xylem. And then you see those rays going through. Those are called, well, they're called rays, and they help with getting um, food and water through the whole tree trunk. We're not going to focus too much on that, but if you've ever wondered what those are when you look at a tree in cross section, that's what those are. And those are just made of parenchyma cells. Okay. Okay. So vascular tissue, part of vascular tissue is xylem. And they're clustered. You'll see a bunch of tracheids and vessels all clustered together. And we'll go through why that is in a bit. Then the other part of vascular tissue that's right near the xylem is called the phloem. And phloem is also super important for the plant because that's how the sugar is getting transported. So after the plant does all that lovely photosynthesis, then it's got to get that sugar that it's making to the rest of the plant body. Especially, when you think about roots that can't do any photosynthesis themselves, you got to make sure that the sugar gets transported to different parts of the body. So we tend to focus in on what we see in angiosperms. That has been the best studied type of phloem. And in the angiosperms, we see sieve tube members. Sometimes people call them sieve tube elements. It's the same thing. Sieve tube members or sieve tube elements. Okay, so now the phloem tissue does not have a secondary cell wall. So that means if they just have a primary cell wall, they're living. They're living cells, so they have cytoplasm. We say they're living but modified because they have given up some of their organelles. So they're modified. And it's interesting that they do this, but it's probably for efficiency of sugar flow. So they're alive, but they're modified. And basically what that causes is that for every sieve tube member, so you see the sieve tube members they're showing are the bigger kind of tannish cells here. And then for every sieve tube member, there's an associated companion cell that they're showing kind of in yellow here, right next to it. So companion cell is named appropriately because it's the companion to the sieve tube member right next to it. All right. And so when I said that the sieve tube members are modified, they've gotten rid of some of their organelles. So what they rely on are those companion cells to help them. So those cells still need to stay alive, get their ATP needs met and all of that. They need their nutrition. So the companion cell actually helps them. They share some of their nutrients with the sieve tube member. And if you experimentally go in and you kill one of those companion cells, you kill the neighboring sieve tube member. That's how dependent the sieve tube member is. And so the companion cell, I like the name, they named it appropriately. It really is the companion and helper to the sieve tube member. You can see the sieve tube members are kind of funky looking. They have what they call sieve plates. Okay. So they stack together and the sieve plates line up with each other end to end to end. Those sieve plates, they look like a sieve. That's why they call them sieve plates. It's all open. It's just opening, open space there. But they stack end to end like that and they match up their sieve plates. And then the sugars and other fluids will flow through there.
If you kill one of the companion cells, will that, obviously you said it'll kill one of the sieve tube members.
Mhm.
Will that like destroy the whole line then because they can't properly transport stuff or not?
Yeah, I could see over time that could disrupt. It could cause, because if that sieve tube member dies, it'll probably hollow out after a bit, and then that would stop the flow. Yeah, yeah. So when I say it's found in more advanced plants, by the way, that's usually we're talking angiosperms because angiosperms are the most advanced plants on our planet. Okay, so it's kind of interesting that this vascular tissue, the xylem is dead, the phloem is living, modified but living, but they're clustered together. You find xylem and phloem near each other all the time. Okay, it's part of the vascular tissue. And so some of the other type of phloem tissue, so we just talked about sieve tube members, which are found in the angiosperms. I just want to mention sieve cells is the second type of phloem tissue. So I do need you to know Sieve tube members are in the angiosperms. Sieve cells are in the more primitive plants. So we say the primitive vascular plants. So we're going to find out later in the semester that there are some plants that have more primitive vascular tissue, and they just have sieve cells instead of sieve tube members. So this was their first crack at phloem tissue. They evolved sieve cells, and it works. We tend to find them in more primitive vascular plants like ferns, okay, and gymnosperms. So when we get into those groups, we'll be talking about sieve cells. There's far less research done on sieve cells. We don't know as much about them compared to sieve tube members, but they're similar in look and, of course, in their function. So getting the sugars throughout the plant body, really, really important. And then if you go all the way to the outside of the plant, we're talking about epidermal tissue. Just, we have epidermal tissue too as our boundary to our outside world. And typically it's one cell thick. Sometimes it's thicker, but usually it's one cell layer thick. And so here you're looking at a leaf in cross section, and they're showing the upper epidermal cells, so that line of cells. And then there's a lower epidermal cell on the bottom of the leaf. Okay. So epidermal cells are typically one cell. Here's a drawing, a little bit more detailed of a leaf. And then the cuticle that's covering the surface there, that's just a layer of wax. Okay, it's a waxy layer. Plants have to cover themselves in wax so that they don't dehydrate, lose water out of their body just randomly. They don't want that, especially because they can't go to the water faucet or the fridge to go get more water. All right, so they have to be super careful about how water leaves their body. They are very protective of their water, and it'll show as we get into this idea here next, which is these areas here. They're called the stoma. Let me go forward a slide here. Stoma is singular. If you just look at one, you'll go, oh, look at that stoma. But usually we use the plural, which is stomata. So the stomata are all of these interesting looking kidney bean shaped cells. So each, you see that there's a pair of these we call them guard cells. Okay. They're paired up like this, and they surround the pore. You can see the pore in this plant is open a little bit. So this must be a pretty well hydrated plant for the pore to be open like that. So the guard cells guard the pore. That's their job. And what happens is those guard cells, if my hands are the guard cells can close over the pore, so the pore is shut down, or it can reveal the pore, and it can be 100% open, 50% open, 10% open. They have exquisite control on how open that pore is, and they do that by controlling the guard cells and whether or not the guard cells will close over the pore or open and reveal the pore. And that's where most of the water leaves their body. The rest of their body, they wax it up and get it very hydrophobic, so water won't leave just through cracks and crevices of the leaf tissue, or even the root tissue is well waxed on the surface. All right, so you see that in this previous drawing. So they've drawn looks like 3 guard cells here with the pores that they're guarding. So we'll talk a little bit more about that. It's fun when you look at it from the surface view. When you look down, you'll do this, I believe. Well, you probably did some of this yesterday where you looked at, but you'll do it again Thursday. So this is a leaf surface, and you can see all those jigsaw shaped, those are just normal epidermal cells. All right. So when you look at a leaf, you have a couple of different types of epidermal cells. You'll have like the normal epidermal cells, and then you'll have the stoma, or the stomata, I should say, that are allowing for, so when that pore is open, what happens is water will start leaving the plant body. The plant can't help that. It's just going to happen. And I'll explain why later. But if you have the pore open, that's great, because then you can take in CO2, so you can do photosynthesis. So when a plant opens its pore, opens its stomata, we typically say, if a plant is opening its stomata, it's usually because it wants to take in CO2. It's just one of the consequences of that is they'll have water leaving their body through that same pore. And so they have to constantly assess and figure out, am I getting dehydrated? And if they are getting dehydrated, then they close down their pore, some or maybe all, so that they can conserve water. Okay. So another thing that you'll see on the epidermis layer are trichomes, depending on the plant species. You tend to see these very commonly on leaves. Stems can have them too, to some extent. And so here you're seeing a very hairy leaf. Someone might say, look at all the hairs on that leaf. The fancy word for that is trichome. So I want you to know that word in a botany class. So trichomes, and they're epidermal cells that have just been elongated and stand up off the leaf. Okay, they're long and thin. And so you're seeing a little bit of them here in this picture on the right, all that tan are the trichomes. And then these funky things here on the lower right, you see them sticking up off the leaf, plus they're glandular. We call those glandular trichomes. If you see something like that, they're usually filled with essential oils. And if you brush your finger or your hand across those leaves, you get usually a very noticeable odor from that essential oil breaking free from the trichomes. So trichomes are important. We'll get into those in more detail when we get into leaves, but they serve several functions for the plant. Defense, one, but they also can slow down the loss of water through the plant body. So we'll talk more about that. Then I said that especially leaves, you see a lot of what we call cuticle. That's just the wax. So here again, it's pink. You can see that this leaf tissue is, well, I guess this is on, well, I think this is leaf, yeah, stoma. It's on a leaf tissue. And so it can be on leaves and stems, branches, but it's just a nice waxy layer to create a hydrophobic covering so that they don't lose water. The only way they're losing water is through the stomata. And it's done for a purpose, but they have to still be careful about it. It can help them, that cuticle, not only with retaining water, but some pathogens have a hard time getting through that depending on how thick it is. So it can help with some protection. And then if you think about when you look at your seeds next week, hopefully what you're going to find when you undo your sandwich, your paper towel sandwich, is you'll see the radical coming out of the seed. And that's what you're seeing here. And you see all those lovely root hairs. So those are epidermal cells too, kind of similar to the trichome idea, where they're just epidermal cells that are long and skinny and stick up off the surface of the root. And so under the microscope, they look like that. And they're just great. So that creates a large surface area. When something like that is long and skinny, that's good surface area. And that really helps them sop up water. So root hairs of the roots are where you see the majority of water uptake. It's really good for water uptake from the soil system. That's why if you've ever read or seen any video on how to repot your plant, they often say, you take your plant from a smaller pot, you put it into a bigger pot, and then they'll say water thoroughly, and check it every day. And the reason they're saying that is no matter how careful you are when you transplant your plant into a fresh pot of soil, you strip most of the root hairs off. They're so fragile. And so they've lost their main way of taking up water. And so you have to provide extra water so that it takes a few days for them to regrow their root hairs. Okay, so those root hairs it's kind of funny. We call them root hairs, but we have to call them trichomes when they're up on the surface. Go figure. All right. Then we have what's called periderm tissues, which we'll get into in more detail. But basically, the stem tissue is the best example of that because as a tree is getting older, it loses its epidermis and replaces it with a periderm. And a periderm is what's going to lead to bark tissue. All right. So a young tree seedling doesn't have that yet, but as it gets older, it'll switch from epidermal cells to a tougher bark system. And we'll learn more about bark when we get into that section on stems. But part of the bark are cork cells. Cork cells are pretty tough and they're dead at maturity. And so it's a tougher way to cover their stem tissue. And then if you're wondering why I have a swimmer here taking a big breath, it's because when you look at stem tissue, sometimes you can see these obvious like breaks. It looks like breaks in the bark. Sometimes you don't see this though, but those are lenticels. Okay, lenticels. And you need to know that term. So lenticels are helping the tree trunk, in this case, breathe. Lenticels, what that is, it's just a region of more loosely packed bark. All right, so it's not so jammed together, one next to the other, all the cells just really perfect against each other. It's just kind of a loose configuration of cells, which means there's a lot of air gaps there, and that allows air to flow into the stem through the lenticels and allow for, because the tree trunk, a lot of those cells are still living and they need air for respiration. They need oxygen. So they have to get a way to get oxygen into the trunk so they can breathe and, so to speak, do respiration. Suberin, by the way, up here, I forgot to say, suberin is another type of wax. We see suberin as part of the bark on tree trunks. Suberin also covers roots below ground. And again, it's just a waxy layer to help waterproof the tissue of the plant. So the lenticels, we'll see those under the microscope a little bit later. It really does help them breathe. It allows for them to get nice access to oxygen. Under the microscope, we'll see this later in the semester. That's what it'll look like. And so you can see it's a loose configuration. And you can imagine oxygen able to get into that area a little bit easier. Okay. Well, we finished that slide set. So the next slide set we got to get into is plant growth, which obviously we'll have to do on Monday, I guess, right? And so that's going to get into a little bit of hormones and how the plants respond to their environment. Okay.
That won't be till Wednesday.
Oh, we don't have class on Monday. Keep forgetting about that. Wow. So we only met twice this week. We'll only meet twice next week. Yikes. Okay. Yes.
Are there no stomata in that area then?
Not really. The lenticel kind of takes the place of it, I guess, if you want to think of it that way. Especially for getting oxygen in.